We're going to move ahead to our second session for today. Next is State of the Science and Research Needs. We have two presenters. After they give their presentation, we may have time for one or two questions. Our first speaker is Jeff Wagner. He is a research scientist at the California Department of Public Health. He received his PhD at the University of North Carolina, Chapel Hill. Go Tar Heels. He's providing subject matter expertise on sampling and analytical methods, study design, and data analysis over there. He also manages a microscopy and special studies laboratory and maintains accredited asbestos analysis programs. Following Jeff will be Charles Wong. He heads the chemistry department at SCWRP, the Southern California Coastal Water Research Project. He received his PhD in civil engineering from the University of Minnesota. He joined SCWRP in 2019, where his research interests included the development of novel methods for quantifying chemicals of emerging concern. Thank you for being with us this morning, gentlemen. Jeff?
Thank you for the introduction. Can everyone hear me and see my full screen?
Looks great, Jeff.
Okay,
yes.
Great. So I'll be speaking today about precision or repeatability of microplastic analyses. And I just want to start with thanking EPA for funding this work. As Harry mentioned, we've been working with EPA for some years now, and to the other labs that participated in the interlaboratory study. So I want to start with the need to understand variability when comparing two different results. For example, the microplastics concentrations at two different wastewater treatment plants. This is separate from the other critical issue that a lot of people have been focusing on recently, which is also critical, accuracy and bias. But this topic deals more with, you can think of them as error bars on your results. And that's really key when you're trying to decide whether two results are really different. So in practice, this variability is a combination of many different sources. And I have sort of a cartoon at the bottom that tries to show the variability that would come from different steps of the sampling and analysis process about a mean, which is what you're trying to actually get at each step. So for example, when you're sampling, you can picture microplastics concentrations at a given location varying over time, and you're grabbing just a grab sample. Another example would be once you got that sample, if it's a big sample, water sample, when you take an aliquot from that sample, you can imagine there would be variability of the concentrations of all the different possible aliquots from that sample about the mean sample concentration. Similarly, when you're preparing the sample, there could be variability from the preparation process. And then finally, the piece that I'm going to be focusing on today is when you analyze a given prep, would repeat analyses of just that prep vary? And some reasons why that might happen is if you choose different fields or particles to analyze each time, there's a certain amount of signal to noise in your spectra for microscopy, but also for other analytical methods that could contribute variability each time you do it. So in regards to field selection for microscopy methods, that pretty much comes from the fact that when you have heavy concentrations of either microplastics or more often of other things besides microplastics, you'll want to, in many cases, spread your deposit out, say on a filter, to minimize the matrix interference and spread out those particles. And in that case, it's quite common to analyze a fraction less than 1 of your sample. It reduces your analysis time, particularly if you have thousands of particles, or if you have a slow instrument. Sometimes it even improves filter flatness and focus issues. But in any case, because of this, there's many different environmental count-based methods, particularly in air samples where this type of practice is assumed. And therefore in the methods and I should add in the accreditations, for these methods, it is always specified that you assess the variability from repeat counts. And I kind of try to show what this looks like for a few of these tests. And asbestos and air, you're actually subsampling the filter and then from there subsampling that subsample with kind of a systematic field pattern. For asbestos in water or air with TM, the actual analyzed area is much smaller. And then on the other extreme, for microplastic analyses, you typically analyze a much greater proportion of your sample. So what we did was set up a precision exercise to try and assess analytical precision from repeat analyses. And this was targeted at infrared instruments. And we prepared both clean water and wastewater reference samples for this exercise. The way we did this was to spike vials of 10 mils of liquid of either ultra-pure water or treated wastewater in methanol to simulate aliquots from larger samples. And we used microplastic spikes from 50 to 500 microns. I should have put on the slide, we used PE, PET, and polystyrene for this exercise. And we designed it so that they were low concentrations, actually less than 40 microplastics per sample. And we did that to accommodate different methods, some of which you might want to count the whole sample and then recognize that every lab would be doing this 7 times for each sample. So this was a worst case for precision, but it may be relevant to cleaner real-world samples. And this was a relatively small exercise with just 4 labs, more of a proof of concept. We did this to test the draft ASTM methods that Harry mentioned earlier, with the acknowledgement that different instruments possess some differences in workflow and the final sample prep, and that performance may depend on these variations. So this is a look at the data flow. Basically, each lab received a spreadsheet template, simple template where you just record the polymers you identified and their sizes. And that had 7 tabs for the 7 duplicates for each sample, and it automatically calculated the relative standard deviation of the counts for each detected polymer type, as well as the total microplastics. We also, as part of the reporting process, asked labs to give some details on their extraction and analysis methods. And just to point out a few relevant differences here, 3 of the 4 labs did filtrations. 1 did a pipette, series of pipettes onto slides. And that resulted in, that was one of the factors that determined how much of the sample they actually prepped for analysis. It ranged from 100%. Most labs actually did 100% for the clean water sample, but much less for the wastewater sample, just because it had so much remaining material in it and it was difficult to deal with. The pipette, the lab that was doing pipetting prepared even less. And as far as that wastewater samples go, yeah, half the labs found it advantageous to do additional chemical treatments to try and reduce that matrix, and the other half did gravity separations. So on the analysis end, they were all IR techniques, and all of them used a microplastic reference spectra in combination with automatic ID. But they varied in the degree of automation. 2 of the labs had automated mapping techniques with autocorrelation maps. Another one did manual particle selection with automated acquisition, and still another one did fully automated particle targeting. And the last thing I'll mention is that one of the labs attempted to reorient the sample for each duplicate, but then that was just for the clean water sample. And I'll show why in this slide. It turned out that that was not a good idea. Even though you may want to assess that as part of your variability analysis, the orange data, or rather the blue data, represents what happened. So basically, you can see that the microplastics counts went down each time, suggesting that there was particle loss from the sample. So that basically introduces an artifact into this precision analysis. So I would propose that an alternative approach would be to just make sure you analyze different fields each time rather than repositioning the sample. And then the orange data is for the wastewater sample. That was where that second process was followed, and you can see you get a much more stable recount over time. I mentioned that the wastewater samples were heavier, and basically because of that, they were much more time intensive for this interlaboratory study. And certainly the degree of automation that can be incorporated into a process helps with that. This is an example of spectral correlation maps. So in this case, you only have one microplastic particle out of hundreds, and these types of correlation maps can help you just find the one microplastic. So it saves time. Moving on to the results, as I mentioned, these were low count spikes. So the labs had detections ranging from roughly 10 to 30 microplastics per sample. So again, fairly low counts. And we instructed the labs to recount all the polymer particles that they found. We knew there would probably be extra polymers besides the spikes in the wastewater, just native to the wastewater, because these were actual wastewater samples. And then there were also extra polymers detected in the clean water sample, probably during the prep process were introduced. But the key, again, for this exercise was not recovery, but reproducibility. So if you see a particle, just see how many times, see if you see it every time. So moving on to the RSDs, they ranged from about 0.1 to 0.6 for the total microplastic counts. And what we found, you can't really see it from this data, but if you plot it, you can see that, as you might expect, those RSDs were dependent on microplastic counts. So this is sort of showing the same data three different ways. But in the upper leftmost graph, you see that solid black curve is what's predicted from Poisson theory, that if you have low counts, your variability just due to random chance in counting will go up. So that implies that, alternatively, that if you want a low RSD, you're going to want to count more microplastic particles. And that is expressed in this equation on the left. 1 over the square root of the mean count is your expected variability due to chance. And what we found was that our data generally followed that trend, although in many cases there was less variability than what would be predicted. So that was interesting. What our main concern was the opposite. Basically, if we would have found that variability was greater, much greater than random chance, that would have suggested additional sources of imprecision in these analyses, or worst case, just analyses methods that you would call unreliable. But fortunately, we found the opposite, that they did follow this Poisson prediction, and in many cases was better. We found a similar result when we did a different Poisson statistics-based analysis. Basically, this is showing the number of trials in which the mean count was obtained. You're never really going to expect all 7 trials to obtain the same mean count. You'll expect sort of a distribution. And again, we found in more cases than not, it was actually better than predicted due to random chance as far as obtaining the mean count. Doing a statistical test at a confidence level of 0.05 showed that 1/3 of the data sets represented truly random recounts. And this is yet another way of showing this data, where a score of 1 means that it was what you would expect for a random recount of low count samples. And then again, the ones that were not representative of random recounts were generally better than what you would expect from a random recount. So in summary, we feel that we did design and complete a successful precision exercise in which the low microplastic counts and wastewater matrix in particular were challenging, though interestingly, we found that the RSDs were similar between the clean water and wastewater matrix. And we found that the RSDs decreased with increasing counts, which suggests that you should either try to aim for a certain threshold number of counts in your analysis to achieve a certain RSD, or if you have to count low counts, scale your expectations accordingly that you will get a higher RSD. That being said, we found RSDs that were similar or lower than random chance, and we think that the automation in these methods likely improved their reproducibility. And then finally, selecting different fields for each duplicate is preferable to reorienting the preps to avoid artifacts. Now, the implications of this exercise, I just wanted to mention, this is just part of the total variability. So additional sampling and subsampling variability assessments are needed. But for just the analytical precision piece alone, I think what could be done in the future is that for a given lab, instrument, method, analyst, and I would argue microplastic range, you can track these things in control charts. So you do it once initially and then update it regularly, say every analysis day, week, or quarter. And you end up with a control chart. I'm showing an example from our lab for our accreditation for asbestos and air. So that generates an RSD that you can report, sort of a characteristic RSD for that lab instrument method analyst that you can put on your reports. It can be multiplied times a given analytical result to give you an analytical uncertainty. And it can also be used as a way of checking real-time your any analytical issues that come up in your lab. So when you do your latest recount, if it's outside of control limits, then you have a reason to investigate problems with your lab. And that's all I have for today. And thank you for having me.
Thank you, Jeff. Next up is Charles.
Pleasure to be here. Thank you all for coming. I was asked to talk a little bit about microplastics analytical development at SCWRP by Scott Coffin, and I'm going to follow up on some of what Scott talked about with regards to our microplastics measurement intercalibration study, which has wrapped up for drinking water and which he has made available as he's mentioned. So what is this? This is basically a study to determine what methods would be suitable for monitoring microplastics in drinking water in response to state legislation mandating this. And this is a significant challenge because up until fairly recently, there hasn't been a whole lot of consensus on how to measure microplastics. And that's problematic because for a bunch of reasons. If you're doing different methods, you could be comparing apples and oranges, and it's hard to intercompare results from data obtained from these methods. It's also not clear what these methods can and cannot do, or if any of these methods are better than others, and also why. And to compound this, it's also not necessarily clear what exactly it is we want to measure with microplastics. Is it their size, their shape, their polymer type? You'll hear more about that in some of the other presentations later on in this webinar. But basically, this is a significant challenge, and to add to this, of course, is that developing these methods often take a long time. You got to figure out what you want. You got to figure out how to go about doing it and what you're after. Then you have to perform various analyses, for example, for a number of different labs to see what the method can do. Look at the results, figure out ways of reducing variability, and then basically repeat the process. So how did we go about doing it for this particular issue, which is to find ways to monitor and measure microplastics in drinking water? Well, the intercalibration study that SCWRP led basically used blind samples and a common playbook, common set of methods that participating labs could basically follow to process and analyze these samples. By blind samples, what I mean is these are samples with known compositions that we've added, but we don't tell the participating laboratories what's in them. So they analyze these as unknowns, and they follow a agreed upon series of procedures to minimize variability from that. And from this, we're basically quantifying a couple of things. We're quantifying accuracy, we're quantifying precision, and we're quantifying feasibility. So accuracy is basically how close can you come to the middle? What are each lab's results compared to what we know is the correct answer based on all sorts of things. Precision is how close can each lab cluster their shots to each other? How repeatable are they with the triplicate samples that they get? And we can parse this in a number of different ways to figure out what these methods can and can't do from the same laboratory, from experienced laboratories, and from labs with different levels of experience, which is important because in monitoring work, there's going to be laboratories that are new to this, and we want to see basically how well they can do and how they can improve if they do need to improve. The final thing, of course, is feasibility by figuring out, well, how much time and effort and energy does it require to do these types of methods? Because that's important too from the monitoring standpoint. So I want to talk a little bit about some highlights from this particular from this exercise and what would fall out from it. Back in 2019, we had a workshop. We invited experts around the world to figure out some candidate methods, which you see here, and also draft some standard operating procedures, again, so that everybody's following the same playbook and we minimize basically people doing their own thing as a source of variability. Previous intercalibration studies have shown that that's a pretty significant source of variability. With regards to the blind samples that we made, here's a little bit of information about them. There were several different types of polymers that were added. These ranged in size all the way down to single-digit micron size to about a millimeter in size. These are broken up into 4 different size fractions, as you see. We have different types of shapes. We've got pellets and fragments and spheres and fibers. And one of the other things we have to simulate what would be in a real sample would be false positives. So many of the particles that you would expect in an environmental sample, including a drinking water sample, aren't going to be microplastics, but they might look like microplastics. So we decided to add material that would basically would be common that you would find, or at least would look like microplastics, and these would include things like sand and pieces of shells and cotton fibers. We even added some bunny fur for my pet bunnies because oddly enough, that looked an awful lot like the actual polymer fibers that we actually did add. So what are some of the matrices? We have clean water. You can see an example from the nice clean water there. That's complete, and that's what I'm going to talk about. We also had environmental matrices such as a proxy for surface water mantitol, the dirty water, which also included algae and sticks and all that sort of stuff. That's that yellow container you see, as well as things like sediment and fish tissue. That data submission is complete, and we are now starting analyses in conjunction with our participating laboratories on these other matrices. So I'm going to confine my remarks to the clean water matrix. Overall, for all 4 matrices, we had 40 participating laboratories in 6 countries. This was a healthy mix of public sector labs at various levels, academic labs and private sector labs, and they ranged from some of the experts in the field since it started to basically labs that had never done this before, with quite a few labs on each particular method. And basically how the lab flow worked was we had blind samples. You can see a bunch of them being ready to ship out that would go to the laboratories. And the laboratories would again follow the playbook of the draft SOPs to process these types of samples and then quantify them using visual microscopy, measure them, figure out how many they are. This is a very common step as currently done in microplastics analysis. What this won't do, of course, is verify what the material is and if it's a plastic particle or something else. So there would be a confirmation step using something like Raman or FTIR spectroscopy or pyro-GCMS. With regards to the clean water matrix, which was a proxy for drinking water, the processing method was basically split them apart, split the size, the particle sizes apart in a number of different sieve sizes, put them on filters, and then basically count and quantify. So what kind of results did we get? These are for some of the candidate methods here. These are box plots showing the distribution of how many particles each of the labs got. That's the mean of those is each of the individual dots. And you see two dotted lines near the top. The lower dotted line is the total number of plastic particles that we actually added. And the top line is the total particles we added, which also included all the false positives. So if you look at this, you kind of go, oh, this doesn't look all that great. More than half the labs got less than half the particles that we actually added, and there's quite a bit of variability involved. But remember, there's also quite a bit of factors under the hood that's we can look at further, and that gives us a lot further insights into what kind of performance these methods have. So what we have plotted here are box plots, and the blue box plot on the left is basically what I showed you in the previous slide, and it's now expressed as relative recovery. But the other box plots now break these down into the 4 different size fractions that we have, the different colors that we have, and the different types of particle shapes that we have. Now you can see there's a lot more going on here. So what you can see here is that with regards to sizes, for anything bigger than about 20 microns or so, as it turns out, the labs actually have pretty good recovery and reasonably tight precision around the actual value. It's the 1 to 20 micron particles, which are very difficult to analyze, and that's known in the literature, that caused all sorts of problems and also was depressing the overall recovery. With regards to things like, let's go to the far right, morphology and the shape, you notice that the labs typically reported a lot more fibers than we actually added. And as it turns out, it's actually very, very easy to contaminate samples because there's particles floating around in the air and on surfaces. So we have to keep things clean and try to do everything you can to minimize contamination. And a lot of the contamination is due to things like fibers. And these would, for example, also include fibers coming from masks from that you would have to wear from the pandemic. With regards to things like color, you can see that things like blue particles tended to be higher. And as it turns out, the blue particles that we did add to these samples tended to be rather fragile and tended to break up. So that's partially what you're seeing there. We can also get quite a bit of information about and insights into best practices. So for example, these plots here show you the recovery for one of the size fractions in terms of both accuracy and precision based on how much experience the labs are reporting. So the blue labs at level 0 have the least amount of experience, and the gray labs at level 2 have the most amount of experience. What we note here is that the least experienced lab tended to overestimate the number of particles that they have, and they tended to be pretty volatile in terms of identifying plastic particles that weren't, and vice versa. As you gain more experience, the recover, you stopped overestimating the number of particles. You started to figure out what a plastic particle looks like and confirm that that is indeed plastic. And basically you weren't all over the map and trying to identify them. So this is good news from the standpoint that once you learn how to do this, you're going to get better, and measurably so. What we also learned is that training and following a clear SOP also helps. So we had a training course back in the fall of 19 where a number of labs went to be trained hands-on on how to do these types of extraction processes and also measure these things. And those labs that did the training got better performance. Also, those labs which followed the draft SOP that we offered, the common playbook, also had better performance than those labs that didn't. And this is important from the standpoint in that a lot of labs doing monitoring aren't going to be familiar with this, but if they are trained on how to do this, and if they have good, clear SOPs to follow, they can get good performance and they can improve their performance. And with regards to spectroscopy for identifying chemicals, as it turns out, both Raman and FTIR, I'm just going to show the Raman data, had really, really good accuracy and were highly accurate at identifying plastic particles from non-plastics and also identifying the specific types of plastic particles. It also turns out that novice labs can also get accurate results. So in other words, once you learn how to use the instrument, you can get good results. And the only real difference between the RUM and FTIR is that FTIR tends to, our data suggested that FTIR tended to do less well for the really, really small particles at 1 to 20 microns, which is consistent with FTIR having fundamental problems due to wavelength issues for particles that are basically that small. Raman tends to do a little bit better with regards to these smaller particles. So what are the things that are falling out from this study? Well, we now understand what these types of methods can and can't do. And this is stuff that's, that Scott has talked about and has posted onto the State Water Board website. It's currently being worked up for, in terms of peer review manuscripts for the journal Chemosphere as a special issue that is dedicated to the study. We also now have SOPs for these methods. These are now refined by the participating labs so that we achieve consensus on what works and what doesn't work and how. And the State Water Board is basically reviewing this. And for all methods, you need to be able to accredit laboratories so that, to show that they can have acceptable performance. We now understand what these, what the performance capabilities are. We know that what a good lab can achieve, and we're now working with eLab to develop the accreditation process for laboratories that would be doing this monitoring work. We have a website that is for all things microplastics in that we're doing, and that's all I've got. And I'd be happy to entertain questions. Thank you for your time.
Thank you, Charles. Thank you, Jeff.